Mitochondrial genetics can be seen in huge medical research studies like antibiotic resistance and cancer progression, but these mighty genetics also give us a better understanding of how cells have evolved over time. Mitochondrial DNA, or mtDNA, is encoded and affected by various evolutionary pressures, some of which can be examined in future studies. In this experiment, we focused on 3 reasons why certain types of core genes stay in mitochondrial DNA as opposed to other mtDNA genes.
Over the course of eukaryotic phylogeny, mitochondria has begun to lose its genomes. Scientists have developed several solutions as to why this is occurring, but they have yet to find and collect the data to support the hypotheses they made. In Module 1, we learned about evolution and how different organisms came about and developed over time. This study is tracing the pieces of qualitative evidence showing the disappearance of genes in the mitochondria. The group of scientists made a system they call hypertraps to track the evolution of mitochondria to find causes of mitochondrial DNA loss. After narrowing down the list of related factors, they connected high guanine cytosine content and high protein hydrophobicity with a pattern of mitochondrial DNA retention and noticed preference of mitochondrially encoded proteins, known as complex centrality.
One reason for certain genes being retained in mtDNA over others is due to the protein complexes that are important for their cells. For example, does this drawing look familiar? Exactly. It is our electron transport chain from earlier in class. One reason for genes is that they're important to ETC protein complexes in eukaryotic cells. This subset of mitochondrial genes has stayed in the organelle throughout its whole evolution, showing the importance of energetic centrality with ETC complexes. A second reason for certain genes being retained in mitochondrial DNA has to do with the nitrogenous bases involved. Think back to our 4 bases: thymine, adenine, cytosine, and guanine. Mitochondrial gene retention across different species is due to guanine cytosine content, or GC content. This is the percentage of nitrogenous bases on a DNA or RNA molecule that are either guanine or cytosine, the strongest bond of the possible bases. As you can see from the picture, there are 3 bonds between cytosine and guanine, whereas only 2 are between thymine and adenine. mtDNA has chemical and physical properties similar to optimal environments that GC-rich nucleic acids survive in. The third and final reason for mtDNA retention comes from hydrophobicity's impact on gene retention, which can be explained by this experiment in particular. In Module 3, we learned about hydrophobicity. It is the physical property of a molecule that is seemingly repelled from a mass of water. This can be seen in our lipid bilayer cell membrane, where polar heads face the water environments and the nonpolar or hydrophobic heads stay on the interior of the cell, avoiding water. Parts of mtDNA that are not hydrophobic will not end up being retained for future generations.
The mitochondria's development can be described by the endosymbiosis theory. A eukaryotic cell engages with an autotrophic alpha proteobacterium cell and completely engulfs the bacteria cell. However, rather than simply digesting the cell, the eukaryotic cell keeps the bacterium as a symbiont. Later on in the process, the symbiont loses some of its own genes and becomes the mitochondria of the newly made cell. This can explain why the mitochondria and chloroplasts, for that matter, have their own set of DNA and genes. However, biologists have discovered that many of the genes that were present at the earliest stages of the mitochondria's biological history cease to exist in eukaryotic cells today. There are multiple explanations for this phenomenon. The first is a familiar theory of natural selection in that it is favorable for the mitochondria to stop producing these genes. If an individual eukaryotic cell with a mutation causing it to have less genes than other cells were to reproduce more abundantly, this could spur on the evolutionary process as natural selection describes. While there is not an abundance of quantitative data to support the current recordings of gene loss in the mitochondria, qualitative analysis ignites discussion in theory as to why the mitochondrial genes are disappearing. Some theories attribute the loss of gene replication to protection against mutations that can harm the mitochondria during gene replication. Others go as far as stating that the mitochondria becomes more energy efficient with less genes in the mtDNA. When noticing this loss in genes, it is also important to note that the majority of the gene retention occurs within the mtDNA. As of now, there is no evidence suggesting why gene retention only occurs there, so we have to assume it is random. However, biologists have theorized the possibility of toxic areas in the cell where gene retention cannot occur or where it will be more detrimental to the area in the cell than beneficial.
The trait of guanine cytosine content, or GC content, and hydrophobicity appeared in more than 97% of data models representative of both R. americana genes and average gene properties across the eukaryotes. The gene retention of GC content and hydrophobicity show a positive correlation. Spearman's rank returns a rho value of 0.72, and regression analysis gives an R-squared value of 0.51. These tests were conducted on an average of genes of both the R. americana and the overall taxa.
As mentioned before, scientists have made some guesses as to why mitochondrial DNA is being lost over time. In this experiment, the scientists looked at the phylogeny of thousands of mitochondrial DNA genomes. The NCBI taxonomy tool was used to create an estimate taxonomic tree of the reference set called Reclinomonas americana. The infer taxonomic tree introduced related mitochondrial properties which were narrowed to GC content, hydrophobicity, and complex centrality. Just as we do in our labs, they used what they already knew and combined it with new information to hypothesize and narrow the possible factors. The HyperTraps tool would create an algorithm that enabled them to create a phylogenetic tree for manipulating the features they chose. The algorithm looks something like this. E1 could lead to event 2 or event 2A. Event 2 could lead to event 3 or event 3A, etc., etc. The probability of these events were recorded. They took the results from the tool to find a pattern and compared it to the information they read in different studies. Equations they developed from the models showed favor of gene retention with high GC content, high hydrophobicity, and complex centrality. So they were right in choosing these features.